The ovum. The ova are developed from the primitive germ cells which are embedded in the substance of the ovaries. Each primitive germ cell gives rise by repeated divisions to a number of smaller cells termed oogonia, from which the ova or primary oocytes are developed. Human ova are extremely minute, measuring about 0.2 mm in diameter and are enclosed within the egg follicles of the ovaries. As a rule, each follicle contains a single ovum, but sometimes two or more are present. By the enlargement and subsequent rupture of a follicle at the surface of the ovary, an ovum is liberated and conveyed by the uterine tube to the cavity of the uterus. Unless it be fertilized, it undergoes no further development and is discharged from the uterus. But if fertilization takes place, it is retained within the uterus and is developed into a new being. In appearance and structure, the ovum differs little from an ordinary cell, but distinctive names have been applied to its several parts. Thus, the cell substance is known as the yolk or oplasm, the nucleus as the germinal vessel, and the nucleolus as a germinal spot. The ovum is enclosed within a thick, transparent envelope, the zona striata or zona pellucida, adhering to the outer surface of which are several layers of cells derived from those of the follicle and collectively constituting the corona radiata. Yolk. The yolk comprises number 1. The cytoplasm of the ordinary animal cell with its spongioplasm and hyaloplasm. This is frequently termed the formative yolk. Number 2. The nutritive yolk or deutoplasm, which consists of numerous rounded granules of fatty and albuminide substances embedded in the cytoplasm. In the mammalian ovum, the nutritive yolk is extremely small in amount and is of service in nourishing the embryo in the early stages of its development only, whereas in the egg of the bird, there is sufficient to supply the chick with nutriment throughout the whole period of incubation. The nutritive yolk not only varies in amount, but in its mode of distribution within the egg. Thus, in some animals it is almost uniformly distributed throughout the cytoplasm. In some, it is centrally placed and is surrounded by cytoplasm. In others, it is accumulated at the lower pole of the ovum, while the cytoplasm occupies the upper pole. A centrosome and centriole are present and lie in the immediate neighborhood of the nucleus. Germinal vessel. The germinal vessel or nucleus is a large spherical body which at first occupies a nearly central position but becomes eccentric as the growth of the ovum proceeds. Its structure is that of an ordinary cell nucleus. It consists of a reticulum or charometon, the meshes of which are filled with charoplasm. While connected with or embedded in the reticulum are a number of chromatin masses and chromosomes, which may present the appearance of a skein or may assume the form of rods or loops. The nucleus is enclosed by a delicate nuclear membrane and contains in its interior a well-defined nucleolus or germinal spot. Coverings of the ovum. The zona striata or zona pellucida is a thick membrane, which under the higher powers of the microscope is seen to be radially striated. It persists for some time after fertilization has occurred and may serve for protection during the earlier stages of segmentation. It is not yet determined whether the zona striata is a product of cytoplasm of the ovum or of the cells of corona radiata or both. The corona radiata consist of 2 or 3 strata of the cells. They are derived from the cells of the follicle and adhere to the outer surface of the zona striata. When the ovum is set free from the follicle, the cells are radially arranged around the zona, those of the innermost layer being columnar in shape. The cells of the corona radiata soon disappear. In some animals they secrete or are replaced by a layer of adhesive protein, which may consist in protecting and nourishing the ovum. The phenomena attending the discharge of the ova from the follicles belong more to the ordinary function of the ovary than to general subject of the embryology, and are therefore described with the anatomy of the ovaries. Maturation of the ovum. Before an ovum can be fertilized, it must undergo a process of maturation or ripening. This takes place previous to or immediately after its escape from the follicles and consists essentially of an unequal subdivision of the ovum, first into 2 and then into 4 cells. 3 of the 4 cells are small, incapable of further development, and are termed polar bodies or podocytes, while the 4th is large and constitutes the mature ovum. The process of maturation has not been observed in the human ovum, but has been carefully studied in the ova of some of the lower animals, to which the following description applies. It was pointed out that the number of chromosomes found in the nucleus is constant for all the cells in an animal of any given species, and that in man the number is probably 24. This applies not only to the somatic cells but to primitive ova and their descendants. For the purpose of illustrating the process of maturation, a species may be taken in which the number of nuclear chromosomes is 4. If an ovum from such be observed at the beginning of the maturation process, it will be seen that the number of its chromosomes is apparently reduced to 2. In reality, however, the number is doubled. Since each chromosome consists of 4 granules grouped to form a tetrad during the metaphase, each tetrad divides into 2 diets, which are equally distributed between the nuclei of the 2 cells formed by the first division of the ovum. One of the cells is almost as large as the original ovum and is named the secondary oocyte. The other is small and is termed the first polar body. The secondary oocyte now undergoes subdivision, during which each diet divides and contributes a single chromosome to the nucleus of each of the 2 resulting cells. This second division is also unequal, producing a large cell which constitutes the mature ovum and a small cell, the second polar body. The first polar body frequently divides while the second is being formed, and as a final result 4 cells are produced, the mature ovum and 3 polar bodies, each of which contains 2 chromosomes, that is 1/2 the number present in the nuclei of the somatic cells of members of the same species. The nucleus of the mature ovum is termed the female pronucleus.